So yesterday what we were studying is regarding the computer aided drug design or drug development, and a related topic is known as a drug discovery pipeline. Pipeline is actually a pathway to the development of a drug. That is the meaning of the word drug discovery pipeline. How a drug is finally produced. For example, some drugs may be naturally present in certain plants. How these plant leaves are finally becoming a capsule. That pathway is what we mean by drug discovery pipeline. So here we will be going through the different steps which are involved in the development of a drug. And to develop a drug, the first important stage is the understanding the disease. We want to treat it COVID, for example, COVID or corona. So you know what is the organism that is causing this COVID. Right now we know that it is virus. So in the case of most of the virus, our antibiotics are not effective. So only medication that only treatment that is presently possible is to use some kind of vaccine, because we have not discovered any antiviral substance that will be able to kill virus. There is a few antiviral drugs are there. For example, acyclovir. Acyclovir is one of the very few antiviral drugs. It is effective against some of the viruses, not all viruses. So other than that, there are no medications available against viral diseases. So the best possibility of treating a viral disease is therefore going for a vaccination. That is why in case of corona, that is what we are doing. And what doctors are normally doing in the case of viral disease is that they do not try to kill the organism. Instead, they try to support the body or whatever symptoms the person is developing, the person is exhibiting at that time. They try to relieve that symptoms in a comfortable manner to the patient. So this is what they are doing. So in the case of any disease, now most of the diseases are presently caused by virus, and because bacterial disease we are somehow better in controlling it. We have antibiotics. So antibiotics are very effective against most of the bacterial disease. So bacterial diseases are not causing much problem for mankind now. But viral disease we do not have any medicine. So we are not presently very effective in controlling viral disease. That is why most of the viral diseases are exhibiting, are becoming a headache. For example, AIDS virus, Ebola virus, COVID virus, all these are actually creating too many problems for mankind. So before any potential new medicine can be discovered, scientists work to understand the disease to be treated and to unravel the underlying cause of the condition. So what happens when a bacteria or a virus is entering your body? How your body is getting affected? What are the major changes the body is exhibiting? These things they have to understand. Here you can see that how they are changing the genes of the patient, how they are affecting the proteins present in the patient, how they are affecting the So this is an important thing that we have to understand. So we have to understand how they are changing the genes, how they are affecting our body, different proteins in your body, and how they are changing the different cells as well as tissues present in your body, and how the disease is finally exhibiting or manifesting in the patient. So this is the series of changes which are happening in the person, and this we have to understand. Only if we know the knowledge, we will be able to treat the patient. So without knowing what is the cause of the disease. For example, the person is showing some kind of symptom. There is a lot of pain. There is everyday cough is there, or there is fever is there, and doctors do not know what is the real cause of the disease. In such case, treating the disease may be very difficult. They will be, instead of giving the right medication, they will be giving some other medication. So this knowledge is the basis for treating any disease or any infection. Now, other second aspect of this drug development is target identification. For example, in the last class we have discovered that there is a biological target. This biological target is what is being influenced by the drug molecule. Okay, so if you do not know which biological target has to be influenced, has to be activated or deactivated, we won't be able to treat. So in the case of every disease, there will be certain important molecules present in our body that has to be controlled in order to treat that disease. For example, if some disease will be affecting liver cell, and if virus is primarily affecting the liver cell, and if we are finding a medicine that will prevent the attachment of virus with the liver cell, then it will be very easy for us to control that disease. So here, in the case of every disease, there will be some biological target. It is on this biological target we will be able to use our drug. So once they have enough understanding of the underlying cause of the disease, the pharmaceutical researchers select a target for potential new medicine. Okay. So that is so we have to first identify the target. So in the case of most of the time, the target will be a protein molecule. Sometimes it can be a gene. So most of the time it will be a protein molecule. So what we are doing is that we will be trying to change or influence this protein molecule. Which protein molecule is responsible for the development of disease? That protein molecule we will have to control. Either we have to activate it or we have to inhibit it. That is how normally the scientists are doing. So it is critical that researchers pick the target that is druggable. That is one that can potentially interact with and be affected by it. So there is an important problem here. We have identified a particular protein, but we know that that protein is responsible for this development of this disease. But we are facing an important problem. We do not find any drug molecule that can influence this protein molecule. So then it will be a big problem. So this is an important situation. The protein molecule that we find that will be responsible for the development of drug should be a druggable, that we should be influenced by a drug. The drug will be able to interact with it. Drug will be able to either activate or inhibit that particular protein molecule. Then only we can call it as a druggable target. So we have to identify the target. Second feature is that we should make sure that this particular target is druggable, which means it can be controlled by a drug. The target can be genes, proteins, RNA, or anything involved in the development of a disease. So this is the second step. One is the understanding the disease. The second step goes to target identification. Now third step is the target validation. Target validation it means we are making sure that this is the particular target, that is, for example, in the use of COVID virus. Right now we know that there is certain kind of coronaviruses. Coronaviruses are very similar to the cold viruses. They are influencing our lungs, they are influencing different organs in your body. So if you know that this is the particular virus that will be influenced with this particular type of cells in our body, then we will have to make sure that our identification of the target is correct. For example, we say that some problem on your head is responsible for this particular disease, and if it is not the right case, then our treatment may go wrong. Instead of, for example, sometimes the real reason may be something affecting your kidney or something affecting your liver. So if you are wrongly identifying the real cause of the disease, then the control. For example, sometimes you go to a certain hospital and there will be a lot of medicine. Nowadays, if you go to a patient with hospital, they will give 10, 20 different types of drugs. And after some time, you may realize that there is no improvement. What is the case? Their identification of the cause of the disease is wrong. If this identification of the cause of the disease is wrong, then treatment may not be effective. So that problem also comes here. We are saying that this particular protein is responsible for the development. For example, you say that protein A. But if the protein A is not correctly responsible for the development of disease, then we are not able to treat the disease with any drug, because the protein C or protein B may be really responsible for that. So that is the problem. So third step is that after identifying the target, we have to make sure that this is the we are going to validate. Validate means we are going to confirm. After choosing a potential target, scientists must show that it is actually involved in disease and can be acted upon by a drug. There are two important conditions. Is that one, we have to make sure that this protein A is the real agent, real culprit responsible for the development of disease. Second thing is that we have to make sure that protein A can be druggable, should be a druggable protein, or druggable means can be influenced by a drug. So target validation is crucial to help scientists avoid research path that look promising but ultimately lead to dead end. This is several times happening in research. What happens is that when we are going for a research, we initially think that this is the real this is the reason for this. This particular protein may be the sole responsible for this particular disease. But after 1 or 2 years, we identify that the protein A that which we have found will be responsible for the disease is not the real agent, not the real culprit. Is some other protein, maybe protein C or protein D may be responsible. So then what happens is that all our money, all our effort, all our time will be wasted. So correct identification is an important thing. The researchers demonstrate that particular target is relevant to disease being studied through complicated experiments in both the living as well as, and so this is why they are testing that there. By studying the action of this particular target in different type of animals as well as in human beings, researchers make sure that their target identification is correct. That process is known as target validation. Then comes to the okay, so we have identified the target. Now what we are going to do is that we have to find a drug. So we have identified that protein A is the target molecule. Then what we are going to do is that we should find some molecule, some drug that can influence the protein A. So that step is known as a drug discovery. So finding a promising molecule, usually a lead compound, that could become a drug. That's the next step in the drug development. Armed with the understanding of the disease, scientists are ready to begin looking for a drug. They search for a molecule or a lead compound that may act on the target to alter the disease course. So protein A is the real agent for the disease. Then we are going to find that anything that can be that will be able to influence this protein A in a inhibiting manner, so that we will be able to suppress the disease. So this step is known as a drug discovery. Sometimes this will take several years. Sometimes we will be able to confirm that protein A is the real agent, but finding the drug that will be able to change or modify protein A is another big task. So if you are not able to find a suitable drug molecule that will be able to influence the protein A, then there is no possibility of curing the disease. So this is also an important stage called drug discovery. Sometimes it may take several years, sometimes decades also it will be taking. So how scientists are normally discovering the drug? There are a number of different pathways in the development of drug. One method is that nature, because nature is full of different types of medicine. We know that there are different types of plants, different types of animals, different types of bacteria, fungi, order, and many of these are giving us a lot of different types of. For example, majority of the antibiotics are obtained from some kind of fungi. So all these are useful in finding out these different type of molecule. Then de novo, thanks to advanced, except we have studied about drug discovery by computer-aided drug discovery. Sometimes we are not able to discover a drug. Then what we are going to do is that with the help of computer-aided drug discovery, computer modeling, we will develop a drug. There is another possibility, that is de novo. De novo. Nova means we are training it fresh. Then mass screening. Mass screening means that with the help of robotics. And for example, now you have a photo of an animal. You can go to the Google and upload that photo in the Google search, image search. The Google will be searching millions of photos available in its database and will pick you the right match. This process is known by the name mass screening. So with the help of computer, this mass screening is now quite easy. So that kind of techniques is possible with the help of modern computational techniques and also robotic techniques. So there is a technique is known as a mass screening. Then we are using biotechnology. What we are doing is that with the help of biotechnology, we will be, for example, there are biotechnologically engineered vaccines are there. Some biotechnology engineered antiviral drugs are there. Sometimes we may not be able to produce certain protein in large quantity in a natural way. Then what we are going to do is that we will be using some biotechnological technique to produce that particular protein in an easier way, either through by injecting it in some plants or in some other animals, so that they will be producing large quantities of the particular drug. So there is also another kind of technology that we are using for the development of a drug. Then we go to the clinical test. In the issue clinical test, there are number of stages are there. The first clinical test will be basically looking at these ADME talks. ADME means absorption, distribution, metabolism, excretion, and toxicology. That is, before a drug is accepted by the government for public release or public distribution, the drug company must conduct this ADME toxicological test. So that means how this drug is absorbed by the body, how the drug is distributed in the body, what happens to drug after taking, then what happens to how the drug will be eliminated from your body, then what are the poisonous effect, harmful effect the drug can cause in our body. All these are coming under the pharmacokinetics. So these aspects of the drug has to be properly studied. So then only government will allow the public usage of this particular drug. So these are number of quantities that will be exhibited by that should be conducted by the scientist to properly evaluate the functioning of the drug. That aspect is known by the name pharmacokinetic. It's a branch of science dealing with how drug is influencing your body. Then lead optimization. Right now you are for example, you are having a fever. What do you do? Most of the time you will be taking a small paracetamol drug. Okay, that is tablet. See, it is a small tablet. That small tablet is capable of reducing your fever after some half an hour or 1 hour. But instead of taking one small tablet, you have to take 1 kilogram of that powder. Then what will be the effect? So it will be nuisance. So what scientists are doing is that they will be improving the quality of the drug, so that even in minor dose, the drug will be highly effective. So normally we are taking some 500 grams of 500 mg of that. That will be enough to treat the patient or reduce the fever in a particular person. But instead of taking the small tablet, if you are to take 250 gram or 500 gram or 1 kilogram of a substance, then that will be creating too much problem. We won't be able to market it properly. So scientists are improving the quality of the substance, improving the efficiency of the drug, and that process is done by the lead optimization. We have identified a certain drug and we are making sure that this particular drug is effective, so that why not improve the quality, so that even in a minor dose this drug can produce a lot of effect. So then only they will be able to market it in the form of a tablet. Otherwise, selling these drugs will be quite difficult. That aspect is actually taken care by a team of biologists will be there, chemistry people will be there. Both will be working together and finally improving the quality as well as efficiency of the drug, so that even in a minor dose the drug will be able to produce significant effect in your body. That aspect is done by the lead optimization. Then we go for preclinical testing. So we will be testing the efficiency of the drug first in animals. For example, we will be trying to test the efficiency of the drug in a rat, then we will be using it in rabbit, then monkey, and finally we will be testing it in human volunteers also. So this step is known as a preclinical test. So lab and animal testing is lab and animal testing to determine if drug is safe enough for human testing. So there are sometimes some drugs may be, for example, you might have noticed that dogs and cats will be eating a lot of things. If we are eating all those things, we will have some kind of stomach infection. So certain medicines which are quite effective in case of lower animals may not be effective in case of higher animals. So that aspect has to be taken into consideration. So in every country there are certain agencies. For example, in the case of USA, it is US Food and Drug Administration, FDA. Here it has National Drug Authority is there in the case of Government of India. They will be looking, for example, if you want to sell a drug, we have discovered a drug, we have to take permission from National Drug Authority. Then only they will be studying the process. We will be submitting a detailed application that we have conducted this much test, we have conducted this much safety clinical test, and all these they will be taking, and they will be also taking to sending it to a team of their experts to make sure that drug is safe for human consumption. This aspect is the preclinical testing, and here you can see that the preclinical testing has three phases. Phase 1 clinical test, in which we are actually testing the drug in a small group of healthy volunteers. Healthy volunteers means they do not have any disease, but they are willing to take that drug, so that the efficiency of the drug, effectiveness of the drug will be. For example, before this corona vaccine is introduced, the scientists are asking for volunteers to make sure that this vaccine is effective. So the first thing is the phase 1 of clinical trial, in which the drug is given to a group of small people, a group of healthy people who are volunteering to take that drug for the benefit of the society. Second is that test in a small group of patients. Why we are there? They do not directly go to the patient first, because if the drug is not effective, the patient may die. So that is why first they test in healthy people. In the second stage, they give the drug to the group of unhealthy patients. Then the phase 3, we are giving the same drug to a large group of patients. So only after the phase 3 trial, in which the drug has been given to thousands of people and felt that the drug is quite effective in treating that particular disease, then that report will be submitted to government, and government will be giving you a drug approval. So this is the new, so we have to apply to the government in that particular country, so that the government, the drug authority or the food and drug administration, and will be studying our application very thoroughly, and they will sometimes they will ask you to modify certain things. So they will be studying mainly the ADMC, toxicological property or pharmacokinetic property, and also the effectiveness of the drug, and they will be also trying to check whether the clinical trials are properly done, or there was some manipulation, some foul play in the clinical trial. All these they will be checking. After that only they will be giving approval to the drug. Then once the drug is approved, then comes the next heading. For example, you are making, for example, if you will, somebody asks you to make a cake, it may be possible for you people to make a cake at home. But if somebody gives you an order to make 10,000 cake next week, is it possible for you? So producing a cake in your home and producing 10,000 cake is completely different. So producing one cake, it's quite easy. With the limited technology, it is possible. Same way, producing a small drug in a lab will be quite easy. But producing the same drug in thousands of in thousand or millions of number may not be easy. That needs completely different technology. Sometimes even when the drug is discovered, this aspect will be suffering. So they may not be able to produce that drug in a large number, large quantity. Then only the drug will be available. If because most of the time this is also a hurdle. Because most of the time this drug industry is controlled by rich people, highly greedy people. Even if the people are dying, they are not going to give any concession. In the case of coronavirus, we have noticed that many of the vaccine company like Madonna, which is a small company, now become billionaire company. They are actually, their owners are billionaires because of they are making huge profit. They were unwilling to give any kind of concession to even poor countries for to treat this corona. So that is how greedy these drug industry people are there. So manufacturing means is completely different thing. So manufacturing means producing something in smaller quantities is also always easy, but making it in larger quantity may not be always possible. So coming to the last aspect, discovery and development of new medicine is a long and a complicated process. Sometimes it takes 10 years, 20 years, 30 years, sometimes more than 50 years. Then each success is built on many prior failures. That happens in drugs industry. So in the development of every drug, so many failures are there. One important aspect of the research is that we have to be capable of accepting failure. Those who are not able to accept failure may not do any research, because most of the time research means 90% failure, 10% success. That happens in every research. Even when India was sending the first satellites into the moon, most of the time these missiles will be coming down and falling on the Bay of Bengal. Even when India was sending the first launch vehicle into the moon, that was a big failure. So after so many failures only, every research succeeds. So that aspect is an important thing. That is why everybody cannot become a scientist. Ability to tolerate, the ability to accept failure and still move on is an important quality any scientist needs. So advancement in understanding the human biology and disease are opening up exciting new possibility for breakthrough in medicine. So this is the idea regarding how the drug is developed, or otherwise known as what are the different steps involved in a drug discovery pipeline. And you can expect this question, and in several question paper I have found this is an essay question.